Good morning, and welcome to the webinar of the Division of Analytical Chemistry of European Chemical Society. I'm Slavica Ražić, current chair of the division. Every two years, we have opportunity to select two exceptional scientists in the field of analytical chemistry in two categories, one for life achievement and another in the mid-career in Europe. This is time now to present you the best among the best, selected by DAC-EuChemS jury. The Division of Analytical Chemistry has established the DAC-EuChemS Award for sustained contribution to analytical chemistry in Europe. The award shall distinguish an European individual who has demonstrated a significant and sustained achievements in analytical chemistry research or education throughout his or her career. This time the DAC award goes to Italy. Dr. Aldo Roda is Professor Emeritus of Analytical Chemistry at the Alma Mater Studiorum, University of Bologna, and Alma Mater Professor and Director of the Analytical and Bioanalytical Laboratory. He has been president of the Division Analytical Chemistry of the Italian Chemical Society from 2003 to 2006. Its vice president from 2010 to 2013, and president for 2 times in the International Society of Bioluminescence and Chemiluminescence. Aldo Roda is vice president of the National Institute of Biosystems and Biostructures and effective member of the Accademia della Scienze dell'Istituto di Bologna. He is co-editor-in-chief of Wiley's journal, Luminescence, the Journal of Biological and Chemical Luminescence, and also associate editor of Elsevier's Biosensors and Bioelectronics. Professor Aldo Roda has published up to 800 publications with H index of 63, with about 14,000 citations and 10 books. His scientific activity is mainly focused on bioanalytical chemistry and biosensors, development of bioanalytical methods, including immunoassays, gene probe assays, and biosensor based on different transduction, mainly chemical luminescence-based principles, miniaturization, lab on a chip, and smartphone-based technology for portable devices, wholesale luminescence biosensors, and bioluminescence resonance energy transfer technology. Separation science is also using liquid chromatography and mass spectrometry for drugs, metabolomics, and proteomics studies. He organized several international conferences. He was a member organizing committee of urinalysis in 1996 in Bologna. He was president of the International Symposium on Bioluminescence and Chemiluminescence Bologna 1998. He was also president of International Symposium on Luminescence Spectrometry, also in Bologna in 2008. He was co-chair of analytical chemistry sessions on IUPAC World Chemistry Congress in Turin 2007. He has been invited to hold the plenary lectures to more than 150 international congresses and symposia. Professor Aldo Roda was founded by many grants from European Union, FP7 capacity in 2014, NATO in 2016, European Space Agency and Bill and Melinda Gates Foundation in 2011. That was a year of chemistry, global year in chemistry, I remember that. He was involved in many teaching activities on basic and advanced analytical chemistry, biosensors, nanoanalytics, and postgraduate courses in PhD in chemistry. He was also supervisor of more than 100 experimental undergraduate and graduate theses. If you add his intense activity at European level in the field of analytical chemistry, the spread of his original ideas, and especially inspiration for young researchers in the field of bioanalytical methods and biosensors based on bio and chemiluminescence, it's really more than obvious how this DAC award is very, very well deserved. It's enough from my side. It's time from lecture. Professor Roda, Aldo, the screen is yours, please.
Good morning to everybody. Thank you very much, Slavica, for the nice detailed presentation. I'm really glad, happy to be voted and to be awarded for this from the jury of the DAC Society. This is a very prestigious award that almost at the end of my career is just I'm very honored to be part of this EuChemS DAC Society group of selected people for that. And I want to thank also the Italian side that they for the nomination for the president of the division of analytical chemistry and the Italian Chemical Society for the nomination. They choose my name among other very distinguished colleagues and scientists. So I'm very particularly happy for this unique and very nice opportunity that I receive today. And let me just start with my presentation. Let me work on put let's see, 1 second. Is okay? You see my
no, Aldo, please, you are sharing the desktop, not the presentation right now.
Now?
That's perfect, thank you.
It's okay? I'm sorry for that. Okay, this is the title of my presentation will be Recent Advances and Challenges in Biochemiluminescence-based biosensor, which has been my field of research since many years. And I still have a good memory of the air analysis 9 to be held in Bologna, organized by the colleague of Bari, Professor Zambonin and Luigi Sabatini, that they did together with we organized a nice event. You can see here, particularly the name of Robert Kellner that was present with me and to have a drink. And then there was an official ceremony for the Sigillum Magnum to distinguish scientists. You can see Walter Duncan and so on. So this is was really a nice event that we had in Bologna. Just a good memory for that. But let me move on my presentation. But you know, of course we need to start from COVID pandemic situation, because you know we are here for the webinar and not in presence for a good air analysis event, just in front to a computer. Because we are affected from this pandemic disaster. And of course, as we know that the rapid and the decentralized diagnostics is very important. They have been demonstrated, and so we need but reliable test of biosensor for accurate diagnosis, for the evolution of the prognosis, for the efficacy of the specific vaccination. We said from the analytical chemistry point of view that the situation is not so magic now. They develop very fast, quickly, many methods, particularly lateral flow, something that were not completely satisfied from the analytical point of view. And this makes some problems in the precise, accurate diagnosis of the disease. So, of course, if people like to have a simple test, fast result is difficult to fit with accuracy and precision. This is the dream of all the scientists, to have something very simple, rapid, but on the meantime, they're supposed to be a reliable test. We are still missing biosensor commercially. We have the glucose, the pregnant test, if you said based on lateral flow, that's all, nothing more. So this is the problem why we don't have any so spread diffusion of a rapid test. why the problem we have is so limited, the diffusion of commercial diffusion, the business diffusion of the biosensor. Because there are limited market demand? No, because the market asks us to develop this method. It's difficult to set up robust rapid test. No way. We are thinking what is the responsible for the lack of advancement. We have trouble with the robustness of the molecular recognition element, but they suffer a little bit for the matrix component. The transduction, which is the best transduction. The competition among fluorescence, reflectors, if you talk about optical or outer Raman nanomaterials, if you talk about optical. So this is a problem that is still open to us. And if we move toward the bio and chemiluminescence, that is my expertise, my part of my field, I want to just to enter in more detail where the bio and chemiluminescent biosensor can be compete with other systems. Which are the particularly bioluminescent feature that makes a choice of those in terms of sensitivity, low cost, chemiluminescent reagent, no matrix effect. So we like just to critically discuss about all these points here in my presentation. Of course, if we are dealing with chemiluminescence, the analytical signal, that means the intensity of the emission of the fluorescence, is related proportional to the yield of the reaction of the chemiluminescence, which is derived from three contributes. The proportion of the reactant of a probe, which is converted in a product, R. The product, the part of the product in excited state, and part is not an excited state, so it's no radiative decay, only this one. And three, how the excited state decay and emit light with the efficiency. So we have to consider all these three stories to, in some way, choose and then identify the biochemiluminescence as a very sensitive test. More the detector. The detector, now we have a quite good advancement in the light detection from PMT, photodiode, CCD, imaging, CMOS. So from the technical point of view, detector is not anymore a problem. But let me just a competition with the other, with the colorimetric, photoluminescent, biochemiluminescent, and thermochemiluminescent. This is quite new stuff from my lab. We talk about sensitivity, simplicity, analytical steps. Of course, we need to have all the three, the best. And you can see colorimetric is okay for simplicity, regionless, photoluminescence, regionless, but it is very complicated to set up a miniaturized system. And then biochemiluminescent is okay for the 2 points, but need to add chemicals. Thermochemiluminescent, we suppose, is our proposal. We propose to have a thermochemiluminescent because we expected to have a sensitive test, simple and regionless. So this is our goal. But let me just for fun, what we did recently with the biosensor, we published a paper, challenges in biosensor. It is better to measure a photon or an electron. So we did this with our friend from Rome, Dalila Mosconi, and that are expert in Fabiano Arduini in electrochemistry. And so we did exactly the same. We used the same region, the same chemicals, antibody, the same technology format, and so on, to have a comparison, really a comparison between the two, the performance of the two way. And we found we found that if we measure hydrogen peroxide, peroxidase, IgG, glucose, you can see the detection limit of this one. They are almost the same, maybe a decade of difference, but nothing so different. So we conclude that the competition between amperometry and chemiluminescence is 1-to-1 or 0-to-0, if you like. So we are still on the same way. So we need to claim that biochemiluminescence is better than the other one, but not often. Let me just talk about in these days what is the recent advancement in biochemiluminescence and biosensor. Actually, we have only two basic systems that have been widely used and commercialized: is the luminol, hydrogen peroxide, peroxidase or catalyst, dioxetane analog, and then in this way we can have develop experience in simple and sensitive form. Bioluminescence take a strong advancement in the molecular biology with the use of natural synthetic luciferase, nano-LUC, nano-bit, active site modification, semi-synthetic luciferin substrate. So we have a strong advancement in bioluminescence too. Thermochemiluminescence is still growing, promising, regionless, and then we talk about something about also this one at the end of my presentation. Let's start, nature, what the nature tell us. The inspired from nature. And all the bioluminescence and chemiluminescence is inspired by nature. If you take the firefly, bacterial, luciferase, bacteria, if you take renilla, reniform, or you take fungi, mushroom recently, they share completely different mechanism. They use different substrate, but they pass to this cyclic peroxide ring. Is always frequent in all of those, frequent. And this is the key point that produce a molecule and in a site state that emit light. So this is familiar, shared by all this. So this dioxyethanol is the ring that is the actor of the, accompanies, is shared by all these different luciferase. But so we copy, we naturally inspire chemiluminescence. So the dioxetane, the luminol, the acridinium ester that are synthetic, that are chemiluminescent molecules, they use the same cyclic ring. Even the luminol, they form this cyclic peroxide to produce a product in an excited state. And this is from the energetic point of view, from different mechanisms that I cannot time to enter in detail, but just that there are nothing magic. From nature, we just transfer and in the synthetic way to mimic. But let me just enter in more detail about the luminol story and dioxetane story, that are the two more important that have been used today to concrete develop biosensor or device. The great discovery was made by my friend. We were together in San Diego with Larry Krishka and Gary Thorpe from England. They found that the addition of enhancer, phenol, iodo-phenol, phenothiazine, due to the reaction, to the classical reaction, great enhance the light emission. Look at that, from the regular luminol with hydrogen peroxide and with the enhancer. They added this enhancement, acting as electron transfer mediator. They found this tremendous and almost steady state, stable for 10, 20 minute signal, light signal. And nowadays, this is the 30-year-old system is still the more sensitive for biosensing. No way, we try to do the better. We collaborate and we design a new enhancer, SPTZ, you can see the name here, was slightly better than the other one, but nothing so magic. We add a co-enhancer that is morpholin, and then we do the better, try to improve the classical enhancer cocktail. But the cocktail was slightly better, but nothing magic, and then people still using this one as the best for imaging, for any kind of application. All the big companies, they still using this one. We, of course, using this enhanced chemiluminescence to develop device, biosensor, using the smartphone technology. The strong advancement in the CMOS technology, in the photo camera of the smartphone is very important, very relevant. So we can measure light coming from a reaction for a probe in which we have a peroxidase labeled. In this case, we use for cortisol, just in case. We use a conventional lateral flow system that has been used for pregnant tests. We modify, we don't use anymore a golden nanoparticle, but we use different labeled in different format, a competitive type or not. And then we design with the 3D printing some cover, demi cover to fit with any kind of smartphone. And then at the end, we were able to have a quantitative evaluation of the test, for instance, to measure cortisol or any, not a plus or minus like it was before. And then this was very happy. And then we were very lucky because we had the way to be founded for a NASA, Italian Space Agency project in which we made a device that is still now in the International Space Station. Of course, we have to measure using the same principle of lateral flow, something that is in microgravity, that is in a really extreme condition. And then so we have to follow all the NASA requirements to confine everything inside, to be sure that the system, the chemistry works and so on. So we made all this device in our lab. And then this has been sent on the International Space Station. And then Paolo Nespoli, that was the Italian astronaut, that in that period was in the International Space Station, did the experiment. And then for us was we are very afraid about to see the experiment in a real time. We followed by the Italian center connected with NASA, Espoli doing the space, the experiment, just briefly 1 minute to see. He just collects saliva with a salivate device. And then it was dressed was Halloween period, like Spider-Man. And then he just follow the indication, collect the oral fluid sample in the mouth, and then put in a syringe. And then we made everything manually, easily, because was the prototype. We had to check, we don't use any electronic device, but just manual, how to mix, to add the reagent and so on. So we did, but for us, you know, we're very anxious to do an experiment in microgravity, how the chemistry works. No doubt that the capillarity of the system works much better here than in land, on Earth. And then he put the saliva sample inside, and mix with the chemicals, the reagent that need for the reaction, everything manually. We can replace with an electronic system, wait for the incubation, and then open the reader. You know, the reader has been done to resist, to be compatible with the 8G gravity. When they go out, they have been very stressed during the way when they reach the International Space Station. So they put inside the cartridge, the reader, and then at the end they measure and we get so we're very, very, very happy about that. It was for years very nice experience for that. Okay, of course, we follow, also, we want to take part of the pandemic story. So our idea, we use the similar technology to measure the serum IgA. That is not IgG as often is measured here, IgM, but is IgA because it is even secreted in saliva. So give us different information. And then we try to improve the quality of the test by adding the way how to mix with the chemiluminescent cocktail that usually move along the strip just to in place directly with something that can place directly on the spot after the reaction. And then in this way, we were able to have a quite excellent result, and we're very happy. But this method is still orphan. We need to make some sponsor that choose our system. Particularly, maybe it's important to use this test to see the vaccination efficacy. This can be maybe a way, but anyway, I don't want to enter in more detail about that, but we also take part of this business. But the most important advancement in the field was the nanomaterial, the use of nanomaterial to replace peroxidase, an enzyme. We don't want anymore an enzyme. We want something that a new catalyst that can be any kind of nano, from nanotube, nanomaterial. And then there have been a plethora, so many publications on the use of nanomaterial in a chemiluminescence system. They use this as a catalyst, as an emitter, an energy donor, energy transfer mechanism, and so on. There are so many. And Hui from China, we did a collaboration with her. She's one of the more distinguished scientists, expert in that field. But until now, if you go in the real world, there are still nothing more efficient than the old 36-old system developed by Larry Krischka. Of course, even the mechanism become complicated. The gold nanoparticle take part of the chemiluminescent reaction. They facilitate the decomposition to produce hydroxy radical, and then to facilitate generation of oxygen radical and luminol radical. So to enter in some way in the mechanism of this reaction. But I don't want to enter in this particular detail because this is still something hypothesized, not really well demonstrated. And at the end of the story, the light emission is nothing so magic respect to the peroxidase. But it's interesting that people are working on it. We work recently, we are working now to replace the gold nanoparticle with octahedral platinum ultra small nanoparticle. We call nanozyme, and then we use, our idea was to take this one and to label an antibody with this nanoparticle, and then we follow the reaction of and we can use this label as in an immunoassay. Then we use a luminol to measure the reaction. We had quite recently satisfactory result in terms of the signal detectability, in terms of the concentration of the probe, but we are still 2 or 3 decades less than the peroxidase enhancer, but it looks promising. And then if you have this kind of label, it's much more robust than an enzyme and easy to control and so on. So it's something that we are working with, but not yet at the end. Nanomaterial as a carrier, there are so many MOFs, CNT, carbon nanotube, nanowire, carbon nanofiber, graphene, and then 2D, and so that has been described. And so many publications in the field, very interesting. Nice, elegant paper, but not yet fully convincing. The people are not yet convinced that this is they give the best performance in respect to the other one. Interesting quantum dots. Quantum dot can we can have when we have the luminol in a excited state, we can have a CRET reaction, chemiluminescent resonant energy transfer to the Q dots. That's very typical acceptor for that. And then you have this one, or you can have a direct reaction with hydrogen peroxide of the Q dot, and then in some way to contribute to enhance the system. This is something very interesting, and we are expecting to have quite more exciting result in the near future about that. Recently, I talked about a few months ago, they use the luminol, they use the simple lateral flow pad antibody labeled with the cobalt iron anti-particle, and this way they said that they were able to achieve a very quantitative signal. And so this is also applied. So the SARS-CoV-2 antigen stimulate a lot of research in the field in the last year, in the last month. But many of these papers has been accepted without to go to the review, to the review trial. But anyway, there are quite exciting news, new proposals. But go back to the other story of the dioxetane story. That is the parallel story. It's also started 35 years ago by my friend Paul Schaap. Paul Schaap did a nice invention in which they have this molecule that is stable. There's a protecting group here. If you remove this group, you can form a phenolate that it can be underwent to a mechanism of decomposition with the formation of an moiety in an excited state with the position light. So if you have something that is able to break, or an ester, for instance, esterase, any kind of, or alkaline phosphatase, you can put something, a phosphate here. You can have the enzyme. The enzyme hydrolyzes this group. They can put slide, and you can measure the activity of the enzyme through the decomposition of the action. This is very interesting. And has been largely used in particularly also in clinical chemistry and also in biosensor development. We had some trouble with the solubility for the efficiency of the system in water, and they had to add surfactant and answer. And more recently, my friend Doron Shabbat from Israel, they start again with the design exploring new territory with dioxetane chemistry. It did a lot of chemistry, a large number of chemistry in which you change the electron withdrawing acidic group inside, the fluorophore and so on. So to make very, very exciting new dioxetane able to be used as even to emitting in the near infrared area, and then very useful for imaging, direct imaging in cells and so on. We use a similar one of the substrate developed by Sharon to measure hydrogen peroxide. But why hydrogen peroxide? That you can use luminol? No, because we want to measure hydrogen peroxide inside to a cell. So you need something that pass across the cell membrane and go inside without to disrupt the cell to measure it. So something so the molecule is quite lipophilic, and so they can enter inside to the cell. Once the hydrogen peroxide is present, we have a formation of intermediate with an unstable compound, and you can see on the slide, and then at the end we have the emission of light. And this is very important, that this decomposition makes and allows us to localize where inside the cell are located the hydrogen peroxide, which is a marker of inflammation and any kind of disease. Okay, but I said, why not to decompose a dioxetane, not chemically, but just with eating the molecule. So we can decompose the dioxetane ethanol molecule by eating at reasonable temperature to produce a ketone in an excited state, and then to measure the light of this one. So we originally designed this kind of molecule. This was the first one that we develop, and then one day they can decompose and they produce this one in a single excited state that emit light. So basically, we need a small device. We have an eating system element to eat like Ito. We have an immobilized antibody, for instance. We can have the reaction, immunological reaction. We eat for a few seconds, decompose the probe, and measure the light. So this is something that, of course, become reagentless. So if we are able to do that, but you know, we did a lot of effort. So many publications recently, and even last week, we have the on Chemo-Photochem, and that in which we modify, taking the technology to push together, pull and push electron donation group to ameliorate, to improve the light efficiency of the system, and then to optimize this probe here. And then we are almost on the way. We have a quite new compound here that I cannot tell you because they are quite new, just a few months ago, and then in which we can have a promising result. Our idea is to label an antibody directly, but we prefer to include this in a nanoparticle, as we did. So we doped the hormosyl silica nanoparticle. We decorated the nanoparticle to be able to bound an antibody. And then we put inside even an energy acceptor. And then we eat the system, and then we measure. But more recently, we said, why not to use, to change, to have not a silica nano container, but to use a smart nano container, which is fluorescent. So we replace the silica nanoparticle with the P dot nanoparticle, the semiconductor P dot nanoparticle, which are fluorescent polymer. So it became the same polymer that made the nanoparticle as the fluorescent energy acceptor. And then to make the FRET reaction and to measure light. So this makes us a very interesting way to make a simple test. The idea was to measure the valproic acid detection with the smartphone technology. We design not a lateral flow, but at a vertical flow format, but following always the immunoreaction. They use a nanoparticle, as I showed before, and then to develop a small cartridge to fix in a smartphone. We use the power supply just to eat, to decompose, and then to measure. We put the cartridge and measure, and we're able to have a quantitative response to measure the therapeutic concentration of the valproic acid. So this we're very quite happy. But let me go back to the bioluminescence story. Bioluminescence story never finish. From the firefly luciferin structure, natural, and the modification of the enzyme, different enzyme, from semi-synthetic nano enzyme, nano-look, and then technology that uses the caged firefly luciferin story. We know since many years that we can play with the luciferin structure to switch the color easily, so to have a multiplexing capability from the blue to the red to infrared now, two with the new one. And then also we can modify the luciferase active side, just modify a single amino acid. We can switch the color easily. So we have a multiple tool, luciferin modification, luciferase modification. Never happy about that. We decide with our friend Bruce Branchini, that is one of the father of the bioluminescence in the state, that he visited me in Bologna, and then we collected the firefly Luciola italica near Bologna in May. That is a small luciferin insect, and then this has been cloned, and then has been expressed in different cells as a mutant, green and red mutant. So to have the flag of the Italian story here is just here. Then we can do more. We can take the the wild type luciferase modified to have a mutant to generate a sort of breath in situ close to the site phenomenon, and any other modification. And this has been done for many papers by Branchini group about this one. But you know, the big effort coming from the Nobel Prize from Osamu Shimura was a friend of mine that worked basically on this equarine, was the father, so he decided equarine with the green fluorescent protein, and they went to bioluminescent resonance energy transfer with the emission of light. This opened new territories for the exploring new hybrid inspired material in which you can have a different luciferase, we have a breath, we have a different receptor, like quantum dot, rod, and so on. So many new possibilities. And so Firefly, Renilla, also many luciferase. We have a nanolook that can be used as a breath, as such, or can be cut into components which are not active when they are separated. They become, when they are closed, they become again active. This from Promega, they did a quite good advancement by Keith Wood. Keith Wood was one of the fellows present with me at the UCSD at the Marie de Luca Lab in 1982. Okay, so we have now quite interesting opportunity. The reported gene technology, in which you can put a reported gene or two reported genes inside to a cell, to yeast cell or any kind of cell, and to measure the production of the protein or the luciferase by the mRNA technology and so on, that is activated by the analyte, interaction with the receptor, and so on. This is very good, but you know, it takes time because the RNA synthesis is a right limiting step. 5 hours, 2 hours, 1 hour, too much. Low dynamic range, but they have a multiplex capability. If you use a different technology in which the analyte can induce a breath between two protein donors that are close together, like a receptor, the receptor can dimerize, you can achieve a breath signal that is short, 1 hour, is a ratiometric signal, wide dynamic range, but is a multiplexing capability. More interesting is, as I showed before, the possibility to have to follow an interaction between two proteins or the analyte antigen immunorecognition element with the two luciferase that are inactive, that become active when they are close together. And then, and this is one recent, very recent talk about the using a development of cell-based luciferase complementary PCA assay for SARS-CoV-2 inhibitor. This is just recently, they use this technology based on this luciferase system. Even another interesting paper coming from the recently, I talked about a few months ago, they use the ACE interaction with the spike S1 of the SARS-CoV-2. The interaction between these two is followed by the interaction between the two nanoluciferase that are not active, but once they are close, they become active. So this is very interesting because you can generate light from nothing inside, no chemical. That's very fascinating technology. But we don't forget the nature. You know, if you go in Brazil following and you follow this railroad worm, that is very interesting. They have an head that is green, is red, and the body green. But they use the same luciferin, the same system, exactly the same, not two different chemicals. The only formation of enolate, of keto oxidase, they are formed two different intermediate excited states that they give an account of the different emission of the two spectra. So this is something that is very fascinating. How we can play with this bioluminescence. We explore other possibility. Why not to take magnetic bioluminescent bacteria, magnetotactic bacteria? There are bacteria that contain inside magnetosome, that they are very interesting. They become magnetic. So we decide to take this one, collaborate with an expert in the field in Germany, and we make this bioluminescent. So we combine the magnetism of the inside of the cell with the light emission, and to use the magnetic property of this bacteria as an actuator for our biosensor. So you take this one, and then you can, with a simple magnetic, just move easily where you like, okay? Where you spit and take in another part. Following this technology, we design a system in which we can move the bacteria from the reaction to the detection chamber area, in which we can easily detect the bacteria, move and stop to the cell, and then we can achieve a quantitative measurement. And this is very interesting because open new way to play with the cell without touching the cell, and then but allowing to separate, to deliver the cell where you like in your system. And finally, what we are doing is just taking, why not to work on multiplexing? Just one example. We can develop a test for toxicity, for instance, that can take account of all the possible toxicity effect of a given chemicals. Or so we can take the bacteria luciferase Fisheri luminescent, spontaneous luminescent, that in blue. Then we take the PP green coli bacteria, that green. Then we take the yeast with the nanolook as a reported gene. Then we take the human egg cell. Again, that's been using with reported orange, PP orange. So we have 4 possibility that we can, if we take a toxic compound like the DBSO and we incubate it, you can see that the light can, in an array that we made, can disappear in presence of high concentration of the toxic. But of course, we need to design a sort of cartridge in which we put all these cells, and we share in the bottom the substrate that does interfere between one with the other one. We use for fun the GoPro aerophile. It's a very using popular camera, very small compact camera, that's very quite good CMOS detector. And then so we made this device procedure to measure the system. And then you can see when for instance, we incubate all this with an antibiotic, with the penicillin, and only the E. coli is affected, and also the other bacteria, also the 2 bacteria strain, the other one are not affected. If you use atrazine, a pesticide that is maybe it play a major role in a human cells. If you use other cell, Vibrio coli, that have been used for toxicology, are not so efficient like this one. So this makes sense to have this one. Interesting is another back to the technology, to the discovery. A large publication on the use of the luciferin, caged luciferin. So you can put a group here in this part of the luciferin, and this become not anymore a substrate for the luciferase. So it's caged. But if you either way to release, to hydrolyze, to remove this group, it become again substrate, and then you can present of the enzyme with a bit of light. So any kind of reaction in this point to introduce any kind of thing like are able to measure activity like caspase, nitroreductase, any kind of activity. And then we can play also to the structure of the luciferin to modify, to optimize the system. We did recently, last couple of months ago, we published a paper in which we develop a continuous air-segmented flow for real-time monitoring of biosalts hydrolase in the colon bacteria. We used many years ago this system in which we have a tube in which the sample move between two bubbles of air, the sample, and enter in the system. And then there is a small peristaltic pump. Now we can use very miniaturized system. Here we can compact in 10 centimeters. We have the detector, and then we have the luciferase enzyme immobilized on nylon coil. So we can put the sample inside. We can manual load or with electro valve, and then we can measure. And then in this way, what do we take? We take the luciferin. We take the aminoluciferin that is still a substrate for luciferase. And we did an amide bound with a bile acid compound here, which is the substrate for the biosalt hydrolase enzyme here. And we put this one. And then when in this one, in presence of the biosalt hydrolase activity, is hydrolyzed and release the luciferin that become uncaged and emit light. And so this is a way to follow and to see in your sample, can be stools or intestinal content, or in a microbiota, you have the presence of biosalt hydrolase activity. And this is a very simple device that can be performed up to 10-20 analyses per hour. Okay, we are almost at the end of my presentation. And this is the, I showed a couple of years ago also the Bill Gates, they show a jar of firefly inside and said, okay, can we use bioluminescence to improve existing technology? It's still something that we need, we trust, and we have. So this is something that we are convinced that there is space to move on biochemiluminescence. But let me conclude to thanks at the beginning of my career, the two mentors of me, this William McElroy, that is he discovered the luciferase and ATP process, and Malin De Luca, that in '92 in Air Lab I was present when the first time the firefly gene was cut and passed in a tobacco plant. So the reported gene technology. And even in that time in California, I had the chance to work with Alan Hoffman. This was a ski competition, but this was in real science. He was my teacher, but he passed away last week. So for me, it was really disappointing. I miss him because he was a great chemist to train me a lot. And then I want to thank my professor, Alberto Breccia, that gave me the opportunity to travel, money, and so on. And more recently, our friend Bruce Branchini and Larry Kriska that here, in which we collaborate for many papers, and also Sylvia Downer from Miami, that we did a lot of work. And finally, my wife, Patrizia Simoni, she did a multiplex work, wife and mother, 3 kids, and then co-workers in my lab. So she was always close to me, supporting my different bad mode sometimes, but positive. And I will thank all of them for attention. Thank you very much.
Thank you, Aldo. It's such an impressive lecture, really. I enjoy so much, and I'm just quite sure that all the audience enjoyed the same, really. So I think it's time now to invite a representative of Springer to join me in this Zoom stage. Please, Nico. Dr. Stefan Pauli was prevented at the very last moment. So we have an excellent replacement, actually. Dr. Nicola Oberbeckman-Winter is a senior editor for chemistry in Springer. Welcome, Nicola. I'm just very happy. I'm very happy that the impressive collection of Professor Roda's works will be complemented with a DAC award for a significant and sustained achievements in analytical chemistry research and education throughout the whole career. And Nicola, you have probably some words to say now. I think it's a moment to share with us. Yeah.
Dear Aldo, dear all, let me just congratulate Lou. It was really a great lecture, and Aldo, it's a very, very well deserved award. Congratulations on behalf of Springer Nature.
Thank you, Nicola. Actually, DAC award certificate is surprisingly already in Bologna. I'm so happy to hear that it was really very fast. But for this occasion, there's a small surprise for you, Aldo. We have a special greeting card for you.
Oh.
Congratulations.
Okay, thank you very much. This is very nice. Yeah, firefly, fire.
Yeah.
We need a fire. Firefly,
Yeah.
firework. You know, that's we are similar. We are close to him. It's light, always light for us.
Yes. Thank you. Congratulations again from all of us. Thank you.
Thank you again.
And now it's time to proceed with the second part of this Awardee webinar organized by Division of Analytical Chemistry. And let me say a few words about this award. DAC has established the Robert Kellner Lecture in memory of the efforts and achievements of the late Professor Robert Kellner from Vienna University of Technology towards the consolidation of analytical chemistry in Europe. This award shall distinguish a person who has made substantial contribution to the advancement of analytical chemistry, research and education. We have one exceptional scientist here today. Professor Karen Falls is coming from University of Strathclyde from Glasgow. She's an outstanding scientist who has created and leads a distinct field of chemistry, namely surface enhanced specially offset resonance Ramen scattering, which has emerged from her world-leading research in the field of surface enhanced Raman spectroscopy. In pioneering this field of scissors, Karen Falls has demonstrated world-leading innovation and originality in her research, as evidenced by the publication of series of high-impact papers in the last 2 years on this topic, whilst maintaining her continued innovation using CES for practical application. Her work on scissors has focused on clinical application to exploit the ability to make molecularly specific measurements, depth to tissue by careful design of the appropriate nanosensors. Also, using multiple label nanoparticles opens up the potential for targeting and detecting multiple cancer biomarkers at depth simultaneously. This will be transformational for clinical applications where targeted nanoparticles could be used to detect and target disease, in particular cancer without the need for biopsy, totally non-invasive. The other major groundbreaking strand of her recent research is in creating new approaches to detect bacteria and bacterial resistance in challenging environments using cells. Initially, this approach was highlighted by detection of meningitis pathogens, where the detection and quantification of 3 different pathogens was successfully achieved by cells for the first time. Her recent work was demonstrated the detection of multiple bacteria in magnetic separation assay that can be used for the detection and quantification of bacteria for both medical diagnostic and food security applications, and in understanding the uptake mechanism of nanoparticles in breast cancer cells. She has an excellent international professional standing within the analytical community and is held in high regard by her peers, as evidenced by multiple awards, including international awards such as the Charles Mann Award from FACS for Applied Raman Spectroscopy and the Craver Award from the Coblenz Society. She has been named 4 times in the Analytical Scientist Power List, Top 50 Women in Analytical Science in 2016, Top 10 Spectroscopists 2018, Top 100 Most Influential Analytical Scientists in 2019. Around the world in 16 scientists 2020, and receives numerous invitations to give plenary talks at leading international conferences. She has been elected a Fellow of the Royal Society of Edinburgh, Scotland's National Academy in 2018, and is elected chair, let me stress this, first female at youngest to date, of UK's Infrared and Raman Discussion Group. She's an elected member of the Analytical Division of the Royal Society of Chemistry as an associate editor for the journal Analyst. This is much more to say about Professor Falls, international leading quality of her research and the significance on the impact of her high professional standing. But this is already enough. It's already clear who is with us today. Enough talking from my side again, and it's time for lecture. Please, Professor Falls, Karen, the screen is yours.
Thank you very much. I'll just try to share this before Are we good? Thank you very much, Savita, for that really lovely introduction, and thank you very much, EuChemS DAC, for choosing me as a recipient of this award. It's a huge honour. Robert Kellner was also a vibrational spectroscopist, and he was a huge advocate for analytical science across Europe, and I'm very honoured to be added to this list of names of colleagues and friends from across Europe. Robert Kellner being a huge advocate of analytical science across Europe, I'm very glad that the UK is still considered part of the analytical science community across Europe post-Brexit. Hopefully, perhaps one day we will again be part of Europe. I would like to start by thanking all my colleagues. I'm receiving this award on the behalf of all their hard work over the years. These are all the scientists that I've had the privilege to work with over the course of my career and who have contributed towards this. In particular, I would like to thank Professor Ewan Smith, who was my PhD supervisor. He retired 15 years ago almost now, but we still work together, and we've published, I think it's over 35 joint publications together, and actually most of these have been after his retirement rather than before. I would also like to thank Duncan Graham, who was originally my postdoc supervisor, but now my close collaborator, friend and colleague. So we have published a lot of papers together and worked together for a very long time now. Both of these people have been my mentors, then my colleagues and my friends over the years, so I would like to thank them. For anyone that is unfamiliar, the University of Strathclyde is in Glasgow in Scotland. We are located right in the centre of the city. This is George Square, and the University of Strathclyde is located, I'm just going to actually put my laser pointer on. The University of Strathclyde is located here right in the heart of the city. We have a huge campus rejuvenation going on with lovely new buildings being built, and our research is located in this Technology Innovation Centre. So please, when you can, come and visit us in Glasgow. So the main area of my research is applying Ramen, in particular citrus enhanced Ramen, to various bioanalytical problems. We generally use a combination of nanoparticles, metal nanoparticles with vibrational spectroscopy. My main drivers, I would say, are sensitivity, so being able to detect molecules, in particular biomolecules, at extremely low concentrations, but also the multiplexing capabilities, which Aldo has discussed in his previous wonderful lecture, where we want to detect as many analytes as possible within one sample. We previously applied this to molecular diagnostics. We have done a lot of work on DNA detection, particularly in my case for DNA related to bacterial infections, and pushing the multiplexing capabilities, where we managed to get to detection of quantitative multiplexing, which was quite a push forward for us. So as well as having multiple analytes in a sample, being also able to quantify how much of each of the analytes are present. We do a lot of work in protein assays, so detection of proteins related to disease. At the moment, we've been working on sepsis biomarkers. We have done a little bit of work in COVID-19, as a lot of us have been doing over the last period, but we have an ongoing project in the detection of drug-induced liver damage. We have previously done a lot of small molecule detection, so drugs, particularly drugs of abuse, as well as explosives, and carrying out nanopatterning approaches on surfaces. We do a lot of work on imaging, both using Ramen and surface-enhanced Ramen, which I'll talk a little bit about today. But also we have a simulated Ramen system here at Strathclyde, which we're really pushing forward to at the moment to look at different disease models. So a lot of you will obviously be familiar with Ramen scattering, but I'm just going to introduce the technique to anyone that's less familiar with it. In Ramen scattering, we use a laser to excite our sample. The majority of that Ramen incident light is just scattered with no change in wavelength, so really scattered. But 1 in a million of those photons, we see a shift in wavelength, and it's this shift in wavelength that we measure, which gives us our nice vibrational fingerprint spectrum. However, the issue with Ramen scattering, it gives us this lovely spectrum, but it's not a very sensitive technique. Only 1 in a million of these photons are Ramen scattered, so it's not a very sensitive approach, although it's very information rich. So what we do is combine Ramen scattering with surface enhancement. So to do this, we absorb our analyte molecule onto a roughened metal surface. And in this case, we predominantly use silver and gold nanoparticles as the enhancing surface. We do less work on solid surfaces. What this does is greatly enhances the Ramen scattering by up to the 10 to the 6. But where possible, we try to exploit the resonance effect as well. So what I mean by this, if this analyte is a coloured molecule, and we tune our excitation wavelength to match the absorbance of that molecule, we greatly increase the scattered light that we get back, and enhancements up to 10 to the 14 have been reported. So this then becomes surface enhanced resonance Ramen, the extra R in here. We're now in a position where our sensitivity is getting similar to that of fluorescence or bioluminescence, as Aldo discussed in the previous lecture. But what we still have, the advantage here is we still have a molecularly specific vibrational fingerprint spectrum rather than the broader band that you would see from fluorescence-based approaches. So we can use these unique peaks in the spectrum to identify molecules, but also components of mixtures. So historically, we have really exploited this multiplexing capability, and what I mean by this is we can use unique labels to give us unique cell spectra at sensitive detection limits to produce a multiplex spectrum that is obviously complex because we have these multiple peaks present, but we then use data analysis approaches to then deconvolute this spectrum to find out what was originally present in the sample. So over the years, we've collaborated particularly with Roy Goodacre at the University of Liverpool and using chemometric-based approaches to both elucidate the contents of a multiplex, but also try and quantify the amount of analyte within that multiplex. We have also worked with Mark Girolami at the University of Cambridge on Bayesian statistics methods to use different approaches to keep deconvolute that data. There's 2 general approaches to SERS, and we have used both approaches. There's either direct detection, where we take our nanoparticles and we absorb the analyte of interest onto the surface. This requires a lot of thought about the surface chemistry. So when you make colloidal nanoparticles, they're highly negatively charged due to the surface area. They have to be to allow them to remain stable in solution. But we need to then somehow absorb our analyte onto that surface, overcoming that negative charge, which is fine if you have a positively charged analyte, but often we have got to manipulate and control the conditions to allow us to absorb molecules on the surface. So historically, we've done a lot of work with DNA. DNA is negatively charged, the nanoparticles are negatively charged, but we can add agents such as sperminin, which is positively charged, to bridge them and bring them together. To We also use indirect approaches, and this is what I'm going to talk only about today, actually, where we effectively use the nanoparticle itself as a label. We can then put specific Ramen reporter molecules, as we term them, but these are dye molecules, which we can tune to be in resonance with our excitation wavelength to get that extra enhancement. And then we can also add biorecognition molecules onto the nanoparticles, such as antibodies, DNA sequences, proteins that will then detect species of interest. So this is the approach that I will talk only about today. So in the first part of the talk, I will talk about one of the nanosensor-based approaches that we're using for the detection of bacterial pathogens. So I'm quite sure I don't need to tell you why it's important to detect bacteria, whether that be in clinical samples or in food samples. It's obviously extremely important to detect which particular bacteria is present and potentially be able to quantify that, because we need to be able to isolate that bacteria or eliminate it. So to know exactly which bacteria is present would allow treatment by the correct antibiotic. And these numbers are very scary, that by 2015, there'll be 10 million deaths from antimicrobial resistance, and a costing global cost of £66 trillion. I guess this is even more frightening to consider on top of the 5.5 million people we have already lost globally in the last 2 years due to the COVID pandemic. So the approaches that can be used, there are some simple biochemical tests, luminescent tests, which generally test for the presence of a bacteria species. They use ATP, for example, and they'll just tell you that there's something there that are less specific. Most of the other approaches, such as microscopy, PCR, ELISA, are very specific, can be very sensitive. However, they usually require a bacterial culture step. So most of these you would need to culture the bacteria usually for at least 24 hours before you carried out these tests. This makes them expensive because you require lab-based equipment, time-consuming because you usually have to include that culture step, and obviously requires specialised equipment and trained staff to carry these out. What that means is that often, so for example, someone presents with a clinical infection, they're usually treated with antibiotics right away, but it could be a delay in the correct antibiotic being administered because it takes time to find out the exact pathogen causing the infection. And what we're actually interested in is detection in the food production area. And currently it can take up to a week to get results back in food production samples because they have to be sent away to external labs, at which point any issue has already been dealt with, but it's unknown what pathogen was present within the environment. So we want to develop faster, simpler approaches that can be used and used in the field. So the approach that we've been using, developed by Haley Cairns, this is, in this case, an E. coli bacteria. So what we have done is we've developed magnetic nanoparticles. These have an iron core and then have a silver layer on the surface. This allows us then to attach biomolecules, biorecognition molecules to the surface, which we can use to attach and detect the bacteria. So in this case, what we have functionalized the magnetic nanoparticle with is CONA, which is a lectin. Lectins bind to the carbohydrates on the surface of bacteria. So they are not specific to one particular bacteria, but we don't want this first step to be specific because we want it to have the potential to detect multiple bacteria. This then allows binding to the bacteria and then magnetic separation to allow us to extract from the sample. We then add our SERS nanoparticle. In this case, it was silver nanoparticles. We have a specific reporter molecule on the surface that gives us a specific SERS response, and a biorecognition molecule. In this case, we have an antibody that is strain and species specific towards the bacteria. So only when both of these nanoparticles bind will we be able to pull down the sample and get a SERS response when the bacteria is present. The reason we use the lectin in that first magnetic pull down step is that these nanoparticles will bind to multiple different strains of bacteria, pulling them out of the sample, and then we can add different SERS nanoparticles, different flavors of nanoparticles, as we tend to say, each of these with a unique Ramen reporter molecule to give a unique response and a different antibody that is specific to a different bacterial strain. We then get a multiplex spectrum, and from this we can deconvolute which bacteria were present based on unique peaks in the spectrum. So this shows the different Ramen reporter molecules that we were using to give us our unique SERS signal. This is the signal we get when the bacteria is present, in this case E. coli, Salmonella and MRSA. We still get a low background signal. It's very hard to wash away all the nanoparticles to get rid of them completely, but we have a huge distinction between the background and sample present. We can simply plot this. If we don't want to look at the full spectrum, we can take the intensity of one peak and just plot the intensity of that in this bar graph, showing the discrimination between our control sample and when the bacteria is present. We are pushing the sensitivity of this approach. So here we have 10,000 colony forming units down to 10, so effectively 10 bacteria. And we can see that the signal decreases, as you would hope, as the concentration of each of the bacteria decreases. And what we find is that we can discriminate clearly between the control and 10 bacteria within the sample. So we're getting down to really good sensitivity, and our recent work is pushing this even lower in the approach that we're using. So we have the sensitivity, but we also want that multiplexing capability. Unfortunately, with bacterial infections, clinical samples, and also bacterial infections, particularly our bacterial presence in food, there is unfortunately often multiple bacterial strains present. So we want to be able to identify all strains that are present within a sample. So we can use the multiplexing capabilities of SERS to do this. This is the individual spectra for each of the bacteria. So again, Salmonella, E. coli, and MRSA. And this is a triplex spectrum when all of them are present. So we can design our reporter molecules such that we see a unique peak that we can use to identify the presence of each bacteria within the spectrum. But equally, we can use data analysis approaches such as PCA. Chemometrics are really required, so it's simple to detect if you have an equivalent amount of each bacteria within the sample, but when it changes and you have a high concentration of one, a low concentration of another, it becomes more challenging to pull this information out of the spectra. So we can use PCA. In this case here, we have the multiplex spectra in the middle and the singleplex spectra here, showing that we can discriminate these within the sample. What we're really pushing towards now is integrating this into a portable format. We are chemists, so I guess we're not always good at taking the assay, the instrument, and then putting them together to make them portable. So we have been working using portable instruments. We've been working with Wasatch Photonics to do this, and we're working on how we integrate the assay into a combined system that we can use at the point of use. Ramen spectrometers are much smaller and much more portable than they used to be when I started out in the game. So thankfully the technology is moving in the way that we want it to do. So we're also using these bio-nano sensor-based approaches for lateral flow-based approaches that Aldo also talked about, where we can use the spectroscopy to try and quantify within the lateral flow rather than just give a yes/no answer as well. So in the second part of the talk, I would like to talk about the work that we've been doing on deep Ramen and imaging approaches. I have quite a lot of acronyms, I realise, in my title. So I want to talk about spatially offset Raman spectroscopy. So conventional Raman, we interrogate the sample and collect from the same point, usually using an objective lens. So the laser goes in and we collect this scattered light at the same position through the same lens normally. And spatially offset Raman, we interrogate the sample with the laser, but then we collect the scattered light at a distance that is offset from the incident light by its distance here. So what that means is we interrogate the sample. It allows us to collect photons from deeper within the sample. So this allows us to use Raman to collect data at deeper depths than you can use using the conventional backscattering approach. As I mentioned at the beginning, Raman is an intrinsically weak process, so you are limited in how deep you can go in the sample because the process and the number of scattered photons is low. So what we want to do is combine this with surface enhanced spatially offset Raman SERS. So if we are able to embed our nanoparticles at depth, then we can hopefully start to obtain signals from much deeper within the sample. So we published the first work on surface enhanced spatially offset Raman over 10 years ago now, with Professor Nick Stone at the University of Exeter and Pavel Matusek at the Rutherford Appleton lab, and with Pavel pioneering the SORS technique. And in our original work, we just did simply embedded some nanoparticles at depth within tissue samples, and we were able to detect through 25 millimeters of tissue, and then in our later work, 5 centimeters. In this case, the nanoparticles were embedded at the center of the sample, so the depth of the nanoparticles in the work that we did was 25 millimeters. And actually, in this original work, although we termed it spatially offset Raman, it was actually using transmission Raman, where the interrogation was carried out from one side of the sample with collection on the other side. And this works well with the work that Nick is doing, because they're interested in detection in breast cancer. In that case, using a mammogram-based approach, the breast would be squashed effectively, and you can use a transmission approach to detect at the other side of the sample. However, we're also interested in doing this using a genuine SORS approach, where you can excite and collect from the same side of the sample. So if you were interrogating here, you wouldn't need to collect at the other side, you can collect from the same side. So we've been using a portable system. This is the first portable SORS system that is developed by Cobalt, which is now owned by Agilent. So this original work was carried out by Faye Nicholson. What we have here is we have an 830 nanometer laser excitation wavelength. We need the redshifted wavelength for it to penetrate through tissue, otherwise the tissue would absorb. We're using large 100 nanometer gold nanoparticles here. So what we hope is that if we can find molecules that are in resonance with this 830 nanometer laser, that we will be able to push the depth that we can detect molecules from by exploiting the surface enhanced resonance from these Raman reporters. So we did some work, a lot of work with the late Mike Detty from the University of Buffalo. Unfortunately, Mike passed away last year, but what he made was these beautiful chalcogen dyes. So these have selenium and sulfur groups. These bind really well to the metal surface, to the gold nanoparticle surface, and they orientate very well on the surface, allowing strong SERS signals to be obtained. So you can see these 3 dye molecules here. The number relates to the absorbance maximum of the dye. So this one absorbs at 676, this one at 823, this one at 959. They have the same structure, they just vary in the length of the chain. Here, the longer this chain is, the more redshifted they are. We also looked at two non-resonant Ramen reporters. These are not coloured molecules, but they give very strong surge response in the near infrared wavelengths. So effectively, what we did is we took some nanoparticles, we put 5 millimeters of tissue in front of the nanoparticles, and then we interrogated through that tissue. And this is the intensity of the main peak in the spectrum. And as we expected, the dye that was in resonance gave us a much greater signal enhancement compared to the non-resonant dyes. And we hope that this will then allow us to detect signals from deeper within the sample. So we then coined the phrase to make it even more complicated acronym, surface enhanced spatially offset resonance Ramen, because we're using the resonance between the laser and the dye. So in our initial simple experiments, we had our portable spectrometer. We have a cuvette here full of nanoparticles, and then we put varying amounts of tissue. In this case, we were just using pork tissue in front of the sample, and then to see how deep we could see these nanoparticles through the tissue. And this response here, the top spectrum is our dye reference, so the signal from the nanoparticles that we're looking for. At the bottom is the spectrum that comes from the tissue itself. And in the middle is our offset spectrum. So we're using an 8 millimeter offset to detect at depth. So through 15 millimeters of tissue in the offset spectrum, you can still very clearly see the signal from the nanoparticles. This is just a subtraction spectrum, subtracting the tissue background from the offset spectrum. So this is a signal from the dye here. And then again, as we pushed this further to 25 millimeters, we could still clearly see the dye signal in the offset spectrum. So we were really encouraged by this because our previous work had been using a benchtop system, and this is using a portable system. So even using this portable system, we were able to locate those nanoparticles buried at a 25 millimeter depth. But in that case, we had a cuvette full of nanoparticles in front of the laser beam. So what happens when we start to reduce the concentration of the nanoparticles? So here we have a bit of pork with some nanoparticles spotted down in the surface, and then with increasing layers of tissue on top of this. What we did at this point was mount the spectrometer. So we have a stage here which we can manually move. And what we wanted to do was create a crude image of where these nanoparticles were located within the tissue by scanning across the surface. Now this is crude imaging because it's having to manually move the stage, and the stage was manually moved by 3 millimeters across the sample and a spectrum taken at each point. But what we discovered was through 15 millimeters of tissue here, we were still able to locate the position of where the nanoparticles were located. This is where nanoparticles are present and they're not. And from this, we were also able to form a false color image map showing the location of the nanoparticles within that tissue. So this gives us hope that we can use an imaging-based approach to locate nanoparticles at depth. However, this was again a spotting of nanoparticles down onto the tissue surface, and wouldn't represent, for example, if you were targeting nanoparticles to a tumour to try and locate that tumour at depth. So we then moved to a multi-cellular tumour spheroid model. So we grew MCF7 breast cancer cells into a spheroid, so effectively starting to mimic the tumour microenvironment by creating a ball of cancer cells. We incubated these with our nanoparticles with our Ramen reporter on it. In this case, we were just using non-specific uptake of the nanoparticles by the tumour by the EPR effect. We then spotted a spheroid down onto the surface of some tissue with spheroid located here, and again put tissue on top of this. Don't ask me what the concentration of nanoparticles was within this system, but it would be very low and perhaps more mimicking what we could to detect if we targeted nanoparticle to the tumour. But effectively, we incubated the nanoparticles into the cells and then grew the spheroid from it, with the nanoparticles then becoming dispersed through the system. So using the imaging approach, we spotted down our spheroid, and then we could image across the surface. And again, we were able to locate the nanoparticles within this system using the peak here. So this gives us an indication that we could potentially use this approach to detect nanoparticles at depth in tumour models. But this original work, we were using a non-targeted approach. We were just using EPR effect to uptake the nanoparticles into the tumour. Later work we've been looking at, we really want to understand the targeting of the nanoparticles to the tumour. Should we use a targeted approach or a non-targeted approach, essentially? And the work that Anastasia was doing, she was using nanoparticles functionalized with specific antibodies that attach to specific phenotypes of cancer, and using this to try and target tumour cells. So what I mean by this, so for example, around 65 to 75% of breast cancers are estrogen positive. They have the estrogen receptor. So Anastasia designed nanoparticles that had a Ramen reporter and attached antibody that was specific to the estrogen receptor on the surface of ER positive cell lines. So taking the ER positive cell line, which is MCF7 breast cancer cells, and an ER negative breast cancer cell line, MDA-MB231, incubating our nanoparticles, with it we found that there was much greater targeting of the specific targeted nanoparticle to the ER positive cell, and there was very little non-specific attachment to the ER negative cell. Now, we would expect some uptake of the nanoparticles by the negative cell line just by non-specific uptake of the nanoparticles, but it was very nice to see that we had much greater nanoparticle uptake and interaction when the nanoparticles were targeted to the receptor. But we did see some non-specific uptake, but we expected for the ER negative cell line. What Anastasia then did was to take the cell and take 3D images through this, so she could map two-dimensional planes, 3D locations throughout that cell. And what we clearly see is the SER signal coming from not just the surface of the cell, but also within the cell, showing us that the nanoparticles are actually being uptaken into the cell. And what this led us to think more about, so are the nanoparticles being uptaken in a receptor-specific manner? Are they binding to the receptor and being uptaken, or is it a non-specific interaction? So just to explain that a bit more, our nanoparticles have a Ramen reporter on it and have an antibody. This antibody will bind to the estrogen receptor on the surface of the nanoparticle. So what we thought from our data is that our nanoparticles are binding to the estrogen receptor and then being internalized through the receptor, forming a vesicle, which then takes the nanoparticles into the system. And our work suggested that this was happening, but also that more nanoparticles were being taken in by this receptor-mediated binding rather than non-specific approaches. So to test this, we blocked the MCF7 cells with excess antibody. So by adding excess antibody, it should bind to the estrogen receptor on the surface of the cells, which means that our nanoparticle can no longer bind, so no longer be uptaken. So any nanoparticles that are then uptaken would not be through receptor-mediated uptake. And what that showed, as we'd hoped, the untreated cells with no antibody blocking the receptor, there was a lot of uptake, but when we blocked the receptor, then the nanoparticles were no longer uptaken. So what that suggests is that we are seeing receptor-mediated uptake of the nanoparticles. And we looked at other approaches as well. So Dinosaur is a clathrin-mediated endocytosis inhibitor. The clathrin is what forms effectively the vesicle that the nanoparticles are taken up. And Dinosaur inhibits dynamin, which is effectively cleaves off that vesicle and allows it to be taken into the cell. So we looked at quite a few different approaches. So we're quite convinced that we think the nanoparticles are being uptaken via the receptor. What we also looked at was fulvestrant. Fulvestrant is a SERD, so it's a drug treatment that is used to treat ER-positive breast cancer. And it has multiple actions, but the action that we were particularly interested in is it actually degrades the estrogen receptor on the surface of the cell. So if it degrades the receptor should be gone. Therefore, if our nanoparticles are uptaken via the receptor, they should not be able to be uptaken anymore. So we incubated our MCF7 cells with the fulvestrant drug, and what you can see here is a Western blot, and this is just showing that as we increase the amount of drug, the amount of receptor decreased on the surface of the cell or within the cell, as you would expect. So the drug does its job, it degrades the estrogen receptor. This is GAP-pH, which is a housekeeping, it's a control, effectively showing that the Western blot working okay. So as we take our control cells with no treatment, we can see a lot of nanoparticle uptake, but as we start to increase the fulvestrant, so in each case we're increasing the amount of receptors that are destroyed in the surface, we see a decreasing uptake of the nanoparticles. And we can track this. So as we increase the fulvestrant, more receptors are destroyed, so we have less uptake of the nanoparticles. And I wouldn't say we can do this quantitatively to get a quantitative response of SERS, which would indicate how much estrogen receptor was on the surface, but it does give a qualitative response, as in, as the number of receptors decrease, the number of nanoparticles uptaken by the cell decreases, and it tracks fairly well what we see in the Western blot. So we get a good correlation between the number of receptors that we can measure by Western blot versus the number of receptors that we see by SERS. So this could be used to monitor drug treatment and the reduction of estrogen receptors as treatment occurs, or potentially the reoccurrence of cancer, or looking at primary cell lines to find out whether a fulvestrant treatment is going to be beneficial for a particular patient. But so far, we haven't done this in vivo, and we really don't want to move to in vivo experiments until we fully understand whether targeted versus non-targeted, and how the nanoparticles work in more realistic systems. But we can already see that targeting, increases the uptake of nanoparticles by the system. So recent work that we've done with Michele and Carla at the University of Strathclyde. Michele is an expert in microfluidics, and he has made these microfluidic chips, which have effectively 1,792 wells in them. In each well, you can grow a spheroid, which means that you can interrogate those spheroids with different reagents, different drugs, to watch how this affects the spheroids within the system. So these are images of the spheroids growing within the well, and how they grow. What we did here is we took two different types of nanoparticle. We have our MCF7 cells, or ER positive cell line. We have a targeted nanoparticle which has the estrogen antibody on it, which has BPE reporter on it, which is a distinctive SERS response. So this is the signal we're effectively looking for. But we had an untargeted control as well, where we have the HER2 receptor on here, which the MCF7 cell line doesn't have, and a different Ramen reporter molecule, which is a distinct signal from here. What this means is we can also track the signal of the untargeted nanoparticle to find out how much non-specific uptake there was by this particle versus the specific uptake by the targeted particle. Here we have our spheroid within the microfluidic chip, and the signal here relates to the estrogen nanoparticle and the unique signal. So this is our specific uptake, targeted, and we still see non-specific uptake, which we would expect to see from the HER2 nanoparticles, but you can see there is a much lower number of these taken up. And this is now a flowing system, so I would expect, as well as some non-specific uptake of untargeted nanoparticles by the EPR effect, you're also flowing nanoparticles at spheroids, so some of them will actually non-specifically just be caught and stuck within that matrix. So it was really good to see that there was a much greater uptake by our targeted nanoparticles versus our non-targeted. So you can see the difference in this darker image here, as well as the overall signal intensity from across the surface. We can also 3D image. This is a very labor-intensive process because the spheroids are quite large, but we can take 2D images at locations through that spheroid to tell us whether the nanoparticles are actually penetrating within the spheroid, into the core of the spheroid, if they're just staying on the surface. And what we saw was we did see penetration of the nanoparticles. So as the nanoparticles are uptaken, not just by the surface of the spheroid, they seem to be migrating into the center of it because we're getting strong signals at locations. And just to try and understand whether we can use this flowing system to try and mimic what would happen in a real system, we also incubated the spheroids with the fulvestrant drug to decrease the ER restrict, well, hopefully reduce the ER receptors in the spheroid model. So this is our control sample showing our nanoparticle uptake. As we increase the amount of fulvestrant, so we saw less effect with 1 micromolar, but when we use 10 micromolar of fulvestrant, it's clearly starting to decrease the amount of estrogen receptors within that spheroid, showing much better, much lower nanoparticle uptake, which we can see here using this bar graph. So it's also created a flowing system that could perhaps allow us to again use cells and potentially primary cells to flow drugs to find out which ones would be the best to treat that particular cancer model. But what we're interested in is taking these components and then moving towards in vivo measurements, where we can then do these measurements at depth. Because initially we need to understand which we can take measurements at depth, but we want to push that depth further. And we now know that targeted nanoparticles have much better uptake than non-targeted. So we really want to fully push this system a bit further before we want to move into in vivo systems, because obviously we want to do as few animal experiments as possible. But that's where we currently are. And hopefully I've shown you an example of the approaches that we're using for detection of disease relevant bacteria in this case, but we are developing other point of use applications that we can do detection within the fields using our nanoparticle based approaches. Where possible, we really want to exploit resonance to get that greater sensitivity in our tumor models and our depth measurement to actually push the depth that we can detect nanoparticles at. And we're really wanting to understand the targeting and location of nanoparticles before we start to move to in vivo molecules. No harming of mice unless we need to. So I'd like to finish. I thanked the wonderful research group that I've had the pleasure of working with over the last 15 years or so of my career. But we also have external collaborators. We collaborate with the DSTL Neil Shand in the development of portable instrumentation. we've worked, I've mentioned Nick and Pavel, and CSORS is going out. We're working with Wasatch in the development of portable instruments. And I talked a bit about the multiplexing. We are also working with a food production company for the detection of bacteria in the food production environment. So I would also like to thank these people without whose funding we couldn't do this research. But most importantly, to thank the people that have been involved in this research over the years. And I will finish there.
Thank you, Karen. This is really great lecture, and I enjoyed it a lot. It's not particularly my research area, but I learned a lot. And it's really a pleasure to listen to all that. So it's again a moment, Nicola, I see you are already here, and I'm very happy that one exceptional scientist will receive the award Robert Kenner Lecture in recognition of her outstanding contribution to analytical sciences in general, and in particular to research in a field of chemistry, namely surface and hair, especially offset resonance, Ramen scattering, and clinical application. Nicola?
What can I add? It would have been much nicer to hand over the certificate in person. Nevertheless, I very much enjoyed your inspiring lecture. So on behalf of Springer Nature, congratulations for receiving this prestigious award by delivering this lecture. Thank you.
Thank you.
Thank you, Nicola. And your special archival award certificate is on the way to Glasgow. And it will arrive very, very soon before 6 p.m. I'm just quite sure. But just for now, again, we have a special gift for you. It's a greeting card for you. Congratulations, Karen.
Thank you very much. I will let you know when the certificate arrives. As Nicola said, it's such a shame that we couldn't actually do this in person, but hopefully, hopefully soon.
I'll be happy for the next occasion when some other awardees will be just already invited. I will just invite you and Aldo also just to join us to repeat the congratulations ceremony.
Oh, that would be wonderful. Thank you.
Yes, and we need that Prosecco wine, what Professor Aldo mentioned, behind the stage.
Yeah.
Thank you, thank you. And let me see if there are any questions in our question box. Marcela, there are not. Okay, so. But this is actually the wording seminar, lecture wording seminar, and it's usually not expected to have many questions, and if any at all. So at the end of this event, closing to the end, I would like just to acknowledge all support we have had in the organization this webinar. And I would just invite all my colleagues in the team just to turn on the video. First of all, DAC-EuChemS jury for excellent work, having in mind many nominations, like we cannot remember how many, so many nominations at now, and strong competition for both awards. Many thanks to DAC-EuChemS jury. EuChemS, Ninera Krastel, as General Secretary, we had so general support from association. To Anna Robinson, administrative assistant who kept a fully control on production of this webinar. Laura Jusset, she is a science communication policy officer. She is not here with us, but he was very responsible and she very, very efficient for external communication announcement and the EuChemS website and social media. For this dark part, I did all that, but for EuChemS, she was the main responsible person. Many thanks to Springer. Dr. Stefan Pauli, who was unfortunately is not with us, editorial director for chemistry in Springer Nature, and Dr. Nicola Ober-Beckman-Winters, senior editor for chemistry in Springer, for a long-term and successful cooperation with DAC. Many thanks to Marcela Alves-Secundo, DAC secretary, who efficiently handled all communication before and during the webinar. Thank you, Marcela, so much. And at the end, an impressive audience of all of you in this Zoom lecture call will be recorded actually, hoping that the next event organized by the Division of Analytical Chemistry of EuChemS will be in a real space in some attractive destination. So on behalf of the whole webinar team, I would like to thank you again and say goodbye, enjoy the afternoon.